Students, further we will study about endosperm. What is endosperm? Endosperm is a nutritive tissue formed for the developing embryo in phanerogames, gymnosperm and angiosperm. In gymnosperm, the endosperm is haploid and formed before fertilization, whereas in angiosperm it is triploid and developed after fertilization. The endosperm in angiosperm is generally a product of three fusion nuclei, that is, one male nucleus and two polar nuclei. The types of endosperm. Endosperm have been divided into three categories on the basis of pattern of formation: nuclear endosperm, cellular endosperm, and heliobial endosperm. Davis in 1966 reported that out of 228 families of angiosperm, nuclear type of endosperm is found in 116 families, cellular type in 72 families, and heliobial type in 17 families. In the nuclear type, the first and the subsequent division of the primary endosperm nucleus are free nuclear division without any immediate cell wall formation. In cellular type, the first and the subsequent nuclear division are accompanied by wall formation, whereas in heliobial type, the first nuclear division is followed by wall formation and the subsequent division are of free nuclear type. And nuclear endosperm. Nuclear endosperm is characteristic by the free nuclear division of the primary endosperm nucleus. The nuclei may remain either free or may become separated by wall formation at a later stage. Usually, first few division of the nuclei are synchronized, that is simultaneously, but in the later stage the nuclei are observed in different stage of division. After the formation of few thousands of free nuclei, these are pushed more and more toward the periphery of the embryo sac due to more and more enlargement of the vacuole. Most of the nuclei become aggregated either toward the micropylar end or toward the chalazal end of the embryo sac. This condition is found in Musa errans, in which some of these nuclei form isolated group and are called nodules. These nodules develop distinct cytoplasmic wall and move to the center of the sac, develop as a separate endospermic mass. After free nuclear division, cell wall formation begins. Normally, the cell wall formation starts from the periphery toward the center or from the apex toward the base. Really, it may start from the chalazal end and proceed toward the micropylar end. As in elatine, the entire embryo sac becomes filled with cell, or the entire endosperm remain free in free nuclear state, expecting one or two layer of the cell toward the periphery. In fasciolus, cell formation may be restricted only in the micropylar part of the sac around the embryo. Cell formation is, however, never found in oxyspora and limnanthes. In coconut, that is Cocos nucifera, is an interesting example of nuclear endosperm. Initially, it undergoes a number of free nuclear division, and by the time it becomes 5 cm long. Clear water containing free nuclei accumulate to the embryo sac. The fluid is now called the liquid syncytium or the milk of endosperm. When the fruit become about 10 cm long, cell wall are formed and each cell enclose several nuclei. So at this stage, the fluid suspended comprises both free nuclei and nuclei enclosed within the cell. Subsequently, layer of cellular endosperms are deposited on the wall of the cavity and is known as the coconut meat. In mature coconut, the liquid endosperm turned milky and now it does not contain free nuclei or cell. Similar type of endosperm is developed is found in Erica catuque. Cellular type of endosperm. In cellular type of endosperm, The division of the primary endosperm nucleus is immediately followed by wall formation. The first division is usually transverse, but it may take place in other plane also. In Adoxa type, the first two divisions of the endosperm mother cell are vertical, which result in the formation of four large identical cylindrical cells. A transverse division is then formed, resulting into 8 cells arranged in two rows. After one more division, transverse division, the further division are irregular and a mass of endospermic cells are formed. Among the family of Aronaceae, Aristolochiaceae and Boraginaceae, the second and the third division are transverse, resulting in a row of 4 or more cells. In Acanthaceae, the primary endosperm nucleus moves toward the chalazal end of the embryo sac and then divide to form two equal cells of two unequal size. The lower smaller cell is called the chalazal chamber, while the bigger one is called the micropylar chamber, which divides and form a linear row of 3 cells. The central cell develops into the endosperm proper, and remaining cell form the micropylar and chalazal haustoria. In the member of Lorenthaceae, a composite endosperm is formed by the fusion of the endosperm of different embryo sac. Since ovule is not found in these plant of the embryo sac of an ovary, comes closer to each other, and during the course of development, the endosperm of different embryo sac fuse and form a composite structure. Cellular or type of endosperm is mainly found in dicotyledon saprophytes and parasite. Example, Cuscuta remain an exception. Thank you.